say that what I'm going to talk about today is rather preliminary. We're still trying to understand some of these results, so I may not be able to answer all your questions. The question is, can we localize differentially expressed genes when we have fully sequenced genomes, and why does it matter? Is this an important question? I think it is. We know that from molecular biological studies that chromatin is organized into functional domains, and that organization does determine patterns of gene expression. And we know this from the classical literature, people that have studied puffing patterns in polygene chromosomes, et cetera, lamp-brush chromosomes in frogs. And of course, from my own interests, could we actually identify the presence of differential patterns of gene expression having to do with the presence of chromosomal inversions, of course, drosophilid, of these guys. So when we look at patterns of puffing in polygene chromosomes like this in Drosophila robusta, we see these areas of dense heterochromatin-like structures which are darkly stained and they're not being transcribed versus the lighter structures in these changes through development. So of course. The species I'm going to talk about today is Drosophila movensis. I'm not going to go through a lot of detail with its background, but we know it uses a number of different host cacti in nature. It has evolved a number of different life histories in response to host plants. It's also diverse patterns of mate recognition, and we now recognize that it's essentially showing signs of incipient speciation in different geographical areas. Its ancestors came from southern Mexico. Its closest relative is Drosophila arizona. Movensis is now located up there. Its host plants, the two major host plants are these guys, and these are the cacti that we use in laboratory experiments. Its favorite host plant, Etges, and you can see these dripping patterns of fermenting cactus juice there where the flies are found. In terms of the biogeography, it's pretty clear that they evolved, were separated in Baja California and reinvented in Maine by switching host plants, which gave rise to differences in host plant adaptation and these different patterns of life histories in Baja California and life history differences in derived populations on the mainland, which is a case of direction I selection. Oh, that's cool. Directional life history evolution. All right, well, this talk is based on the fact that we do have a sequenced genome. It's a work in progress, and this is just some of the details from this. It's partially annotated because of the work with melanogaster, but we have done a number of different microarray experiments, and this is what this talk is going to be based on. So this experiment, or one that we've done, involves two populations from each of the geographical regions which we reared in the laboratory on fermented cactus. We did expression studies for groups from males and females, both virgin and unmated, and there are 4 replications, so this is 128 total microarray experiment. And this is the problem. When you design these wonderful experiments, only when you see the results do you realize what kind of real trouble you're in. Because these are the results from this experiment in terms of the numbers of differentially expressed genes. And as you can see, without even guessing too much, most of the variations in the interaction terms, which I find to be the most interesting part of this. So the question is, where are these genes located? Now, and so for these major treatment effects and interactions, these are the ones we focused on. Now remember, I said inversions were important in this story. There are inversions on the second and third chromosomes, and Drosophila movensis has been known for a very long time. One of Bill Heath's students did his thesis on this. And these are homologous to 3R and 2L Drosophila melanogaster. So there are 6 chromosomes of movensis. So this is the kind of data that I've been looking at. These are the mapped locations for all of the genes that show significant gene expression due to sex. for the 6 chromosomes. It shows this pattern of the sort of demasculinization of the X, which was published a while ago in Drosophilid melanogaster, and then you see most of the significant, the numbers of dots below essentially are a female-based increases in gene expression versus male. But if you look at all of those, statistically, almost all of the chromosomes show this pattern, which I don't think is particularly important, I guess. We don't know what this is. For the interaction terms, these are all of the significant differences for sex by cactus mapped according to their physical location. This box down here in the 3rd chromosome is the known location for the 3rd chromosome inversion in this species. We do not know what the breakpoints are yet for the 2nd chromosome inversion. And this is another one, exon 20 test. The question here, which occurred to me, are these differentially expressed genes clustered or not? Are they just simply a random pattern across the chromosomes? Or are there groups of them that we can actually localize, which might have something to do with their functional significance as groups of coordinately expressed genes? How do we do this? Well, the problem is, what's the underlying distribution that you're testing against? The underlying distribution is the physical location of the genes. All right, so how do you take lists of differentially expressed genes in their location and compare them with the actual physical location of known genes across each of the chromosomes and come up with a statistical test and say that there are significant clusters of differentially expressed genes given the genes in that neighborhood? And to my surprise, I found that there's very little information on this in the literature. A colleague of mine, one of the co-authors of this, suggested so-called runs test of dichotomized data. It basically tests to see if the number of runs in a sequence, and this will be the differentially or not differentially expressed genes, is lower or higher than expected by change. Okay, so just go look up this in so-called roll, and they'll tell you how this works. And so we did this for slotting windows of megabase overlapped by 250 kilobases. It results in this so-called T statistic from so-called Rolf, which they defined as the number of runs either being fewer or greater. It's normally distributed with large samples, which we have, so this is good. And the ratio of differentially expressed runs over the non-DE runs, if that's over significantly greater than 1, then there are more differentially expressed groups of genes in an area than there are non-differentially. And this is exactly what we're looking for. My colleague also calculated the ratio of the median length of DE versus non-DE runs. Okay, and this also is an indication that differentially expressed genes in groups are significantly clustered in some regions of the genome, which is what we're looking for. So here's a result from the runs test. So these are the chromosomal physical structural pictures of the 6 chromosomes, 2, 3, 4, 5, 6, and the X chromosome, where the gray boxes here indicate the numbers of where you find higher median run length, indication of clustering. And the black line is the results of the runs test for DE and non-differentially expressed genes. And by resampling, he was able to generate both false discovery rate p-value expectations for both 0.05 and 0.0005 for being really conservative. And as you can see, for the most part, there's almost none of these blackened lines that get out of the FTR region, which means there's not very much evidence for clustering for sex differences. This is the result for the ratio between differential and non-differential run lengths, and he was able to, again, resample this, so the pink regions are the 95% confidence intervals, and then you can see there are some close cases where they seem to be outside of the 95% confidence intervals, but for the most part, they're all contained within. So there's, again, there's not much overt evidence statistically that, at least using these methods, that there's significant gene clustering across all of the chromosomes. And in the end, if we use all of these tests as filters, the results are that they're essentially 2 treatment factors, sex by cactus and sex by mating status, whether they're virgin or mated, where there were significant numbers of clusters in certain windows along the chromosome, and numbers of genes that were differentially expressed in those windows. Okay? So the answer is, there's something here that we can work with. So here's the chromosome 2 cluster out there on the right end up there, enclosed by the red bars. The blue lines there, the dotted lines, that's the physical location of the 3rd chromosome inversion. So that's the actual physical location of where this cluster is on the 2nd chromosome. And for the sex times mating interaction term, there are 3 clusters, 2 of them on the 2nd, and 1 of them on the X. So this is great, okay? This is telling us that we've localized groups of genes that are essentially coordinately overexpressed, underexpressed, okay, on these chromosomes, which is exactly what I want. So, what do you do then? You do functional enrichment, you use gene ontology programs, we use David, which has now just been updated. And the idea here is we can generate lists of genes in these neighborhoods. We can actually try to find out what they do according to their orthology with melanogastric genes. And then we can actually see if they cluster in some sort of functional group. So for the one on the top there, as you can see, well anyway, over there, the Go term over there with the 53 genes, they're all related to chromosomal condensation. Okay, so we need to go look at and see what those genes actually are, what they do. The more interesting one is on the 2nd chromosome for sex times mating. There seems to be an overrepresentation of lipid metabolism genes, which of course are very interesting for a number of reasons. And on the X chromosome, there are no significant gene clusters that were recovered by David, but there are lots of genes, and they have diverse functions. Cuticle proteins, some oxidoreductases, chorion genes, of course, which is associated with mating status, of course, and olfactory receptor genes. So this is, to my joy and satisfaction, a statistical way that I don't think has been employed before to actually show that you can actually localize differentially expressed groups of genes that are of some functional importance because of the way we did these experiments, and we know the great history of the species and its ecology and biogeography. We can actually interpret this in some sort of functional sense. Okay. Thank you. That's the next step, is to see whether different chromosomes have similar groups of genes that are differentially expressed. Again, you don't know for the most part what the 3-dimensional orientation of chromosomes are in most eukaryotes. In Drosophila, there's a chromocenter. These are all telecentric chromosomes, and they're completed. So they're basically fused at one end, which is called, the centric. They're not metacentric chromosomes. What that actually means in terms of gene expression is, I don't think is known. There's a lot of interesting papers that are coming out, if you're interested in this, in the literature, which are showing how the 3-dimensional structure of the chromatin actually is important in terms of gene expression. I think it's really. Yes?
So when you say sex by mating status, I take it you're talking about the mating status of both the male and the female.
Yes.
I mean, a lot of people just see the virgin females and then think about
Right, we did.
Yeah. So I'm wondering if you have a feel for whether the difference is happening with the male or the female that's making that interaction. Or both.
Well, most of them were, most of
The females are changing, I guess.
You'd have to actually, this is the grand challenge of doing these sorts of things, because you actually have to look at the actual structure of the interaction terms. In other words, so in females that are virgins and females that are mated, how do they differ with, or the other way around with males?
Yeah.
And you have to look at the groups of genes that differ in those contrasts. It's very time consuming, but that's what exactly what
That's what you're going for.
that's what we've already actually done. Yeah, that's exactly.
All right, we got a coffee hour now. Anybody wants to